Hello. In this module, I'm going to talk about the modern concept of gene. I'll also be talking about the discovery of nucleic acid. So let us see what is this modern concept of a gene. We once again define gene as a segment of DNA which is responsible for the inheritance and expression of that character. Is that correct? That is what is definition of gene. But here in the year 1955, Seymour Benzer introduced something called as the modern concept of a gene. He basically used these terms called as cistron, muton and recon for what was formerly called as gene. So now we won't call it gene anymore. We'll either call it cistron or we'll call it muton or we will call it recon depending on what function it does. Okay, and once again, please remember these terms were given by Seymour Benzer. Okay, fine. Let us see these three terms individually. First, let's look at this DNA. Okay, a small segment of this DNA, which was formerly called as gene, is known to form RNA. And this RNA will then give you protein. That is how protein is formed, correct? That is from RNA, which is made from the DNA. Yes. Now this segment of DNA, which was formerly called as gene, will now get a new name. So let us see a segment of DNA having information for the synthesis of a particular protein and functions as a hereditary unit is now called as cistron. So gene or cistron are two terms that can now be used interchangeably. Okay, so what was formerly called as gene is now called as cistron. So you can say that cistron is a segment of DNA which has the information for the synthesis of protein. Cistron can also be called as unit of function, and if you look at its size, it can range to several hundred base pairs long. Okay, now let's look at the next term that Seymour Benzer introduced. Once again, what you see here is DNA, and now you see here are two sets of genes. Okay, now see what is happening. In the first set, the color is changing to yellow, in the next is changing to green. Now remember, this change in the DNA is called as mutation. But now this segment of DNA that undergoes mutation will be specifically called as muton. Okay, so what is muton? Muton is the segment of DNA that undergoes mutation, correct? And let me tell you that muton is also called as unit of mutation. And if you look at its size, it could be as small as one base pair to about a few base pairs long. Fine. Now let's look at this third one. Okay, once again you see the DNA here. Okay, you see that small area right there, which would be called as gene originally. Fine. Now I'm looking at a pair of it, right? And here I'm going to show the same pair of gene. Okay, now see what is happening. Crossing over will take place and the genes will get exchanged, right? So now this segment of DNA that participates in recombination through crossing over during meiosis is going to be called as a recon. So recon is the unit of recombination. Okay, by size, if you look, recons could be about few to many base pairs long, right? So these are the three new terms, namely cistron, muton and recon, that were introduced by Seymour Benzer, and this is called as the modern concept of a gene. Let's look ahead to this man. He is Friedrich Miescher. He is known to discover nucleic acid. And how did he do that? So in 1869, Friedrich Miescher separated a cellular substance from the nuclei of pus cells. And because it came from the nuclei, he called it nuclein. Later on, it was seen that nuclein had acidic properties. And because it had acidic properties, the name was changed to nucleic acid. In our cell, there are two different types of nucleic acid. And the two nucleic acids, namely, are, obviously you know this one, deoxyribonucleic acid, and the other one is ribonucleic acid. So the two types of nucleic acid which is found in most cells are deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. Okay, this brings us to the end of the module where I have introduced the modern concept of a gene. Also, I have just given you the introduction to the discovery of nucleic acid. In the next module, I'm going to look at the chemical components of nucleic acid. Thank you.